We have three experienced researchers and grant writers and study section members to talk to you about kind of the inside scoop, what goes on inside NIH and other study sections, and how you can avoid common mistakes and pitfalls. At the far end of the table, we have John Christman, who's a professor and pulmonologist in the Department of Internal Medicine, who's been long time funded by the NIH and has also served on multiple study sections, including 2 T32s and the National Heart, Lung, and Blood Institute, and also involved in some international study sections I was interested, like the Austrian Science Foundation. And then we have Chandan Sin, who is a professor in the Department of Surgery and the director of the Center for Regenerative Medicine and Cell-Based Therapies, and also the director of the CCTS Innovation Program. He also has a long list of NIH-funded and also other government and foundation-funded research, and been involved in many study sections as well. And then finally, Janice Kiecolt-Glaser, who is the director of the Institute for Behavior Medicine Research and a professor of psychiatry in the College of Medicine. She's been funded extensively by the National Cancer Institute, but other NIH institutes as well. And again, has also sat on many study sections. And so thank you guys for coming to us. And I'm just going to, I guess we'll take turns talking about some of your insights and maybe have answer questions from the audience as well.
Yeah, just to add to that, so I'm a physician scientist, and I have been funded by the VA for 25 years until I moved here, and by the NIH for almost 25 years. Sometimes I was funded by both, obviously some overlap. I've been on at least 30 to 40 study sections, if you count all the ones on the phone, both SAT, LIR, the LCMI, lots of things like that. So I have a lot of experience. I also was associate editor of 3 of our big journals for a collective amount of 16 years. So I've got a lot of experience. I'm just one good piece of news. The grant that I put in March got a percentile at 9%, so I don't have to give up my lab after so long, and I was really happy about that. I have another one pending. So that's where my perspective is as we get a little further along. So maybe the other two could say a little bit more about their backgrounds.
Yeah, so I have been NIH funded for the last 20 years since I took over as the assistant professor way back in California. And I hold VA funding, DoD funding, all types of funding, and also have chaired the SAT study section, surgery and anesthesia trauma, and now serve on several other emphasis panels, have visited institutions for certain types of grants, and also serve on some international. So I think taken together, there's significant sort of exposure to this system. I've also been the editor-in-chief of a journal for the last 19 years, handled thousands of manuscripts. So I think together it all adds up. So hopefully we can give you some good advice.
Mm-hmm. I've been funded by NIH since, I think, 1987, so as a PI. So I've dealt with a lot of reviews. I've reviewed a lot of grants. I've served on a couple of different panels as a full-time member, a lot of ad hocs. And I'm currently a member of the NCCIH Council.
So just to add a couple of things to kind of get this started, I jotted a few things down. One thing is that I think it is important to have statistics. It doesn't matter what kind of research you do, whether it's big data, small data, clinical trials, comparative effectiveness, epidemiology, outcomes research, or if you're working in addition on a molecular target. It's very important to have a statistician, and as an editor, I'll often say that when a paper doesn't seem to have one, should include a statistician at the level of authorship. And I think that's really important for some of the reasons that were just discussed by the last speaker. The biggest problem that I see with helping first-time grantors, whether it's a K award or an R01, is just waiting to the last minute. If you give it to someone to look at and there's 2 weeks to go. I mean, that seems like a lot of time to you, but with a lot of other things on your plate, it's hard to look at, and all you can do is some damage control at that time. You can't really enrich the science that much, so you really need a lot of time. It takes me 2 to 3 months to write a new grant, and there's no easy way around it. It's really slow, but a lot of times I'm working with new investigators that will not have it'll all be in their head, but they won't really have it done and on paper until just a few weeks before. So start early. You really want to plan out and get key supportive data. You don't have to have a lot of data. Theoretically, new investigators don't need data at all, but if you have it, and you should have it, it should be important. The other things that are kind of missed, I think, and just a little bit overview of this. One is you need to really make sure you've got strong institutional support. And that might mean writing a draft for your professor or whoever your department chair is, and really putting down some nice things and strong support. They can modify it, of course, and they will, but don't depend on them to put everything down in a really strong way. Significance is really important, and remember, it's the National Institute of Human Health, not the National Institute of Mouse Health. So, you might be studying something other than humans, but if it's the NIH, it's really should be important to health. The last speaker talked a little bit about this inductive, unbiased approach, and it's a nice thing to do to get do RNA-seq and discover something, but that doesn't fly too well in a new grant, especially for a young investigator. It didn't fly too well for me as an established investigator. And then the last two things I want to say is it really helps to identify and meet and talk to your program officer at the NIH. They're nice people, they're paid to help you, they want to help you, they want you to be successful, and they can really even look over your specific aim page and other things. And then the second thing is be persistent. I mean, I'll tell you, I don't remember ever really failing at anything until I started doing research and had rejected grants. And when I left Vanderbilt after 16 years, I threw away a whole bookcase of grants that never got funded. And it was refreshing to do that. And I had remembered these grants as being so wonderful. But when I looked at them, I thought, I got to get rid of these. I want to just censor this for the rest of my life. And I did. So be persistent. You do get better. It is a learned skill. So those are the sort of initial comments I have.
Okay. All right, so many things you're going to hear is going to be more or less sound similar, but I'm going to try to make it a little bit different, knowing what is just covered. So I think in a grant writing strategy, as you just heard, for a mid or a senior investigator is very different from an early stage investigator. And to be very honest, as I see it, there are people at the study section waiting to fund you. And you have to make sure you don't give them a reason not to fund you. And that's exactly how it is. Trust me. People feel sad when they score you poorly because you did everything else good and then missed out on a really important thing, and all of a sudden your score dropped. And there are people, that then feel bad about it. So they are truly there to help you out. Now, how do you build a case? And this, is my personal view, and I think as we discuss, you're going to see a more general view. So my view is that, as a peer reviewer, I'd like to know, who I'm funding. So the NIH bias sketch, you really need to put some time in it. It's not the unimportant part of the grant, especially if you're an ESI. I think when all this preliminary data is unfolding and you're still wondering, should I go this way or that way, that's a good time to put a lot of time and thought into your personal statement, into the areas of an expertise that you're going to claim that is yours. And I believe that it should not be in a copy-paste of something else that was written for somebody else or for some other grant. You really try to make all those areas of expertise converge and make the case in favor of what you're proposing. And along those lines, it's very important also to consider what would you pick to be the topic of your grant. So most of the time, if you're working in a lab, my general rule of thumb is don't try, if you're working in a larger environment with some well-known PI around you and stuff, try to make sure that you're not proposing something that is can be viewed as an incremental development over what that parent PI is already doing. Usually in a lab, there are different types of data. Try not to take the data that is in a straight line with everything else that is happening with the lab. There's oftentimes findings that really depart from the central theme of that lab, and maybe that would be your pick. Because the overall idea is you have to really defend that we need another champion of that area. If there are already a lot of champions doing very well, it's very difficult to get in there and say, me too. And usually that doesn't score well. But sometimes, and I'm not suggesting you do so, but sometimes things that go against the grain a little bit, that yes, this is how it is known, but by the way, if you go to a more relevant model, that's not really how it works. Okay? For that, you don't need a major discovery. You're sort of, in a very thorough and rigorous manner, challenging what is already known, and people would immediately see a big need for your lab, that this is what mainstream science is saying. Now, this person using this technique has been able to see now something that does not fit the common notion. And that, I think, interests people. Some other things that needs to be borne in mind is, say you are about to propose what we would call a paradigm-shifting major innovation. Now, a lot of the times I see that people are very skeptic about accepting major innovation from an ESI. So sometimes, as I suggest to people in our group, that you may have to go publish that in a reputed journal and actually show that it has passed muster, and then utilize that as your base to claim further work in that area. But if you just show preliminary data that is claiming something substantial, you would think that, well, this is really innovative. People would question. But I don't see this in my lab, and I've been doing this for 15 years. And those types of comments come. And then all of a sudden, the 27 other people that don't even work in that area will say, well, if John is saying so, well, I will side with John. If he didn't see, I wouldn't have seen it either. So maybe this new investigator is not doing it right. So sometimes, if it is really sort of on the highly innovative side, it is worth having a high-quality paper, not some, but really high-quality paper, even a short communication, just to get the idea out, because that would go through a rigorous peer review process. They'll let you do revisions, so on and so forth, and that's then out. Coming back to the proposal now, so I couldn't emphasize enough how important is your resume, because, when I get a stack of grants, I think I spend very little time in essentially, deciding what is to be triaged, what I don't want to read. And then I spend a lot of time on very few grants that look very exciting. And that's, sort of human nature. If you find like a very flawed and sloppily written stuff, you want to set it aside. And then essentially after that, you're looking for reasons to be written to triage it, because the decision is already made and now you're just writing it up. But then you're in this stack, people really want to read all of it carefully. So you have to be in this stack and not in that stack. And for that, I would believe, even if you think, well, aesthetics doesn't matter or not, I think it does matter. If it is sloppy in presenting, maybe you're also sloppy in your science. So, like John said, I would think that while you're writing the grant, try to have a group of people that will advise you, around you, throughout the development process of the grant. I would even say the last 6 months, of that journey. And then ping pong with them. Then after it's all done, then you have enough time. And then, so there's no point of hastily submitting, because you have only so many chances. And also, please remember that once you do really bad, and then you again come back hastily with another really bad, people start remembering your names. Oh, this person again came back. Oh, I saw that last spring. Oh, they are back again. You don't want to have that reputation. So I don't think that going in hastily, just because somebody above you is pressurizing that this cycle you must submit, you really have to resist that. That's a part of being a funded investigator. How to resist submitting something that is not ready is a part of the art. I think I'll just stop there so we hear Jan, and then we can go into a conversation mode. Yeah.
I want to echo the first speaker as well. The biostatisticians are really critical. I have seen so many grants basically fall because there was not reasonable biostatistical expertise. A friend of mine recently got funded the first round through. She had excellent statistical expertise, but they didn't, even though she had that in her credentials, she's a psychologist. She got a biostatistician on the grant, and it fared better. She didn't get the same criticisms. So I think it really matters just having the expertise as part of your grant. If you don't have a key element of the grant in your portfolio, something you've already demonstrated, get a collaborator. For example, if you're proposing a clinical trial and you've never run a clinical trial, have someone as a co-I who's run clinical trials, because it really does matter. People, I've seen time and time again, people will say, well, they're a junior investigator, they've never run a clinical trial. Having someone as a co-I on the grant will make a difference, who will talk about in their biosketch their experience with their clinical trials. And if their biosketch doesn't have it, send it back and ask that they put it in, because that's the reason you wanted them in there in the first place. Some kind of odd comments, but take a look at your study section membership before you submit for the sitting members. You want to know who's there. And if it's someone who is likely to disagree with you, then it's a key person in the area, then you need to make sure your arguments are very clear, because that person could really torpedo you. So I typically will scan the section, and sometimes I'll do a quick lit search on the folks just to see, what am I missing, who do I not know? Are there people I better watch out for in terms of who I cite and don't cite? Because, again, if it's a big gun and you're just proposing something in their area and you don't cite them at all, well, there may be some feedback for that. Know the section's issues. Different sections have different pet peeves that they have. I was talking with one of my ex-fellows recently about two sections he'd been sitting on and what their pet peeves were. One called MESH, he said, what I knew already is it's run by what we would call the sleep mafia. Sleep is really central to what they do and to what many people on the section do. So even if your grant has nothing to do with sleep on that section, the sleep mafia will want to know how you're going to address the issue. And that really varies by section, so ask someone who's been on the section. When you respond to reviews, don't be combative. You may have a reviewer for an idiot. It doesn't matter. I had one grant where I was criticized because benign control group was not appropriate for this project. I didn't have a benign control group. I had a within subjects design. So in the response, I said, I was puzzled by this comment since there is no benign control group. I wasn't going to say, you idiot, there's no benign control group. I did, however, email the SRO and say, after I got the review, and say, here is a reason that I could appeal. I'm not going to, but I think you should know about this reviewer's accuracy as well. And when I submitted, I sent her a note and said, I know you can't guarantee reviewers, but I just want to remind you about this reviewer in my grant. And then finally, when I first started writing grants, I didn't have any models for grant writing at the time that were in my field. You really need a good model. How did someone set it up? How do they express things? How do they set up their aims? A good model is invaluable. It doesn't have to be what you're doing, but something you can understand enough to see what they're doing in terms of the flow and how that actually matters.
So being a little self-effacing, I'll tell you this secret. So the first grant I wrote was to the American Heart Association, and in those days they didn't triage, they reviewed every grant, and mine was at the 99.9th percentile, which means that out of 1,000 grants, mine was the worst one. I had never actually seen a grant before. I had no idea how to write one. I just had nothing. But, you know, I took a lot of different courses in grant writing, and I did it over and over and over again. And, you know, ultimately I got better at it. The thing that I had the hardest time learning was, so unlike many of you, I never actually had much in the way of formal training and research. I sort of learned it through a whole variety of people and going to conferences and meetings and thinking, and I was able to actually assimilate that over time, and I've published now over 150 papers that are all mostly cell and molecular biology, very few, in fact, no real clinical patient papers, in spite of the fact that I'm a critical care specialist and I just take care of sick patients all the time. But what I tried to do was impress the reviewers by, I thought if I could understand it, it must not be complicated enough or sophisticated enough. So I would get reviews back that would say something like, you know, given an army with unlimited resources, overambitious. I can't tell you how many of the reviews said that before I finally calmed down and really started thinking concisely and straight, and you know, I had a lot of help from a lot of people, and I think that's you know, one lesson that I learned really the hard way that you guys should really think about very early before you put in that first grant. If it should be reasonably easy for somebody to understand exactly what you're doing, and it ought to be important. You need to think big, as Chandan was saying too.
A few additional comments. You know, first, I cannot emphasize enough how important it is to have the look of the grant the right way. So you don't want to, I know you have a lot of thoughts in your mind, you don't want to pack everything there. Some seminal experiments need to be packed. And then under additional alternative, you can give the hint of what else you might be doing. And for your preliminary data, your publication list, and based on your primary line of investigation, people will start believing that you could. So not every experiment and every detail. Then what happens is the primary line of investigation is not clear, and there are questions about, and also people are reading a lot at that time, so make the reading experience easy for them. Give appropriate space, you know, very important. Now a few things that have recently come up in the last couple of years that needs attention. One is, repeatedly I'm seeing that, you know, by gender. So whatever you're saying, is there a chance this would split by gender? So process that in your mind. In some cases it would be more applicable, in some cases less applicable, but gender is something that folks, you know. Then if you're talking about, say, somebody studying, say, stroke, okay? Now stroke does not happen to a juvenile population that's perfectly healthy. Maybe it does, but that's an exception, not the rule. So what would happen if you have these other cardiovascular profile, risk profiles high? So the context of the disease, people are now talking more about that. Should you be really doing this in a mouse that is 8-week-old, as most experiments are done, or should you take these animals further into their age and make sure that some of these risk, you know, goes up, and then you do, that is very important. And the other set of important criteria based on which score does matter would be rigor and reproducibility. So you heard a little bit from the statistician about that, but you have to be really very transparent about the way you've collected your data, and then what really is your control. And is it a sham? It's a mock, site design, the control, and then when the question changes, how something else becomes a control, and the reagents you're using, have they been validated? So there's a section now for that. Now they say that reagent authentication validation piece should not factor into the score, but I can tell you it does, because if you're not using validated reagents, people score you poorly on approach, which is within the main. So because that could decide the direction of your findings and therefore your line of thinking. So if there is a reagent that the next door lab generated, make sure you put some time in getting the confidence of your peers that this reagent is really measuring what you're exactly supposed to measure, and not some very, very vague, maybe in that area there is very little in a reagent available, and this is the best, but it's really not that specific. That too you have to call out. As long as you call it out, and that's the only reagent available, it will not be held against you. But you have to recognize the limitations and make sure your interpretation is informed by that recognition.
There's a story that I've heard that I think really summarizes many of the key issues you hear in terms of new investigators on study sections. It's about how God applied for a grant on creation to NIH and was triaged. So he called his program officer, and he said, "How can this be? I'm God." And so the program officer said, "Well, let's go through your pink sheet and see what it says." Well, first of all, it seems like it's a little diffuse. You're proposing all these things about trees and animals and people. You're not focused. It's just so much that it leaves people confused and not clear about what your major objective is. The second thing is that your timeline seems unrealistic. You're talking about getting all this work done in a week. And that, we have really experienced scientists, and they really thought that they couldn't do that amount of work in a week, so that you may be a bit overambitious. But the final thing, I think, really is a very serious flaw. You don't have any published articles. All you have is one chapter in a book.
That's funny. How about we have 15 minutes more? How about if you guys sort of take over and lead the questions and see how the panel responds, because we want to individualize this as much as possible.
Yeah.
My name is Zaibo Li from pathology. So my question is, how many specific aims do you recommend in a grant proposal, usually?
Well, I've been telling everybody that it's easier to write 2 than 3. I mean, I'll just tell you, from the grants that I'm asked to review, that third one is usually neglected, and then it's a target for reviewers. 2 well done. One of my faculty is writing a grant right now, and he has 3 specific aims because he thought he needed 3. And it turns out 1 and 2 are really just different ways of looking at the same thing, and they could be combined. People used to say that 1 would be done in cells, 2 in animals, and 3 in humans, but that's really too much for 1 grant. So,
yeah, if you ask me, I would say 3. Unless you're doing, especially if you're somebody new, and it leaves you enough, and the 3 should not be connected. So they have their own lines of inquiry. So even if 1 fails, the other 2 have a fair chance of getting succeeded. But if you are doing heavily in humans, and then there are budget limitations, then 2 makes sense, because human studies need to be done in a given way, no compromise. That would be a good answer.
Mm-hmm.
And part of what I tend to do is, on the third aim, it's often labeled clearly exploratory aim. And it may be a way that I put in things that I would not, that I need to have in there, that I know I would get hit on. For example, in a recent grant, I talked about sex differences as an exploratory aim. We weren't powered for sex differences, so we really couldn't see them necessarily. But at least to say it was an exploratory aim, since it was going to be a reviewer target if it wasn't mentioned, may be helpful.
Yeah, I think anything small and practical is good. Yeah, go ahead.
Yeah, I was just going to ask a little bit deeper. Do you think that they'll ding you for doing exploratory? Because that's the one, I like the concept, because I think you're always kind of proposing for this next part, but I'm always a little nervous to do it, just so they're going to say, why don't you stick with 1 and 2? Because is it really necessary?
Personally, I typically have 1 that's exploratory, and it's worked. I don't know how that works in other sections and other fields.
Yeah, in our types of business, they try to make sure that you do the exploration before you, provide, or come to the hypotheses. And so it doesn't usually score well unless you're talking about R21 or R03 in the R01 mechanism or the K99R00 mechanism that like you to do the exploration behind the scenes and come, but there are exceptions to that.
Yeah, I kind of think about specific aims as papers. I try to think of how many papers I want to put in a specific aim. What will I publish out of this? So, I'm writing a specific aim that might have 4 different sets of experiments that would result in 4 different papers. Specific aim 2 might be another 4 papers or that sort of thing is the way I've conceived of it, because I want the reviewers to go away and say, this is going to be publishable data. And usually I don't try to speculate on something I might do if I don't have supportive data for it. That hasn't worked well for me.
So if God did not have any publications, so how do, I've never been successful with any grant. So you're either dinged because you're not successful in your field with enough publications, or you do not have a track record of having grants. When does the first break actually come in? Because you tag along with someone, but you're not the PI. So, from your experience, when do you actually crack the first? It's so hard. It seems it's getting harder. It's too. I can't.
Yeah.
I'm in a very similar God situation.
That's a tough one.
So, I can just give you, how we would do in our place. So anybody that is in the rank of a senior postdoc or research scientist, I've often had discussions with them that they're now in a particular line of investigation that is mine, but certain offshoots, say we ran a proteomic or some omic, and they are free to take a piece and call it theirs, and my name will never appear in that. And this is a fair discussion to have. Unless you try to do it without telling the PI, then the PI may feel not so good about it, but the PI is there to help you out. And if you carve it out in a way that doesn't really wedge into what the PI's mainstream work is, and nowadays there's so much omic data, that if you really mine it properly and do a few other preliminary studies, and that's how, and then you all of a sudden become the last author, the grad student working with you is the first author or something, and now you have your first paper. That happens with everybody that's worked with me. I think that's essentially how they took off.
It really helps to sort of mine what grants are out there. There are a lot of small grants. Having small grants is highly predictive of academic success. So, you might not get a K award right away, you might not get a R01 right away. It is a big wall to climb. It's very steep to do that. But getting a AHA, American Heart Association, American Lung Association, American Thoracic Society, Parker B. Francis, those are the kinds of things. Cystic Fibrosis Foundation, they've got a lot of money. Those are the kind of things in my field that are amenable to younger investigators with limited data and experience. But it is very important to have some publications. I mean, I don't think you'll ever get a grant unless you've got some publications. So how many do you need? I don't know, 3, 4, 5, something like that. Almost all of us are able to do that through our fellowship or our postdoc or during our PhD. That's what you need is some publications. First author, and some middle author. Middle author is a nice mark of somebody who can network well. And I really look for that. What were they involved in, not just what did they push and do themselves.
So I think technology is speeding ahead of us all the time, and I always get nervous in proposing in a grant using some type of newer technology, but then again recognize that it's incredibly powerful for some of it. What do you guys see on study sections for using newer technologies that maybe are somewhat validated but not higher level utilization? Have you guys seen them on your study sections at all?
Innovation is one of the things that is looked for in NIH grants, one of the 5 scorable criterion, and that can be technical, it can be scientific, it can there's lots of different ways to call innovation, but using the best piece of equipment and getting the best data that's been validated and is the highest level people are looking for. They don't want you to use older techniques that aren't necessarily as accurate. Almost all of the sophisticated techniques now have run as core facilities with very expert directors, and it helps to get a little data. And I think just about every university on the face of the earth will let early stage and new investigators get a free bit of data to kind of seed the well, so to speak.
So I would echo what John said, and also echo a little bit what Jan said a few moments ago, is that say you're working on a particular new technology, you try to see that one of the key experts of the technology is also your co-I. So if they know that if you run into trouble, you have somebody next to you. Now, if there's a technology that you are developing, that's a completely separate story, because that will then have to go through a lot of, and I would recommend in those cases that is a technology born in your lab, that you publish, get some level of acceptance so people know that this is reliable, not just rely on preliminary data and say, well, I can change the world with this technology, and there's zero publication, and that's a bad place to be. Mm-hmm. Yeah.
Yeah. So in the last grant I wrote, the one that just got the 9 percentile, we're looking at a very unique isoform of a histone deacetylase, and it was reported by a very good investigator in Norway, and I searched him down, finally found him on holiday in northern Norway, and he wrote a nice letter to say that he would assist me, and actually provided us with some novel plasmids. And that's the kind of thing you need to do. When I wrote a VA grant one time, I wanted P50 knockout mice because I was doing work on NF-kappa B, and I ended up calling David Baltimore, and his secretary let me through, and he wrote me a letter for my VA merit review. So, you need to go, worldwide, whatever it is. Scientists love helping each other, and if there's somebody out there, just go for it. Again, think big. But align yourself with the best there is. That's one good piece of advice.
Questions over here?
I really did do that, by the way.
Good for you.
I know innovation is a big, as you mentioned, is a part of the scoring. Do you have any other advice on how to talk about innovation? Because I know there's concern that if you're too innovative, they won't fund you, but it's this balance of you've got kind of risk-averse study sections, but they also want innovation.
Let you guys do that.
Okay. I think when you're claiming innovation and also significance, it's better to be very pointed about exactly what you're claiming you're innovating. So to really lay the background succinctly and say where it stops, what is the void, and what exact void are you filling. Sometimes if you write it in very broad strokes, people are going to say, this is not, and I know better than that, this is already done, I don't know what she's claiming or he's claiming. Don't fall into that trap. So it's very important to exactly paint where it stops, the state of the art, and what is the void, why is it an important void, paint that void, and then fill in what you're filling in. That way and that applies also to significance. So many people die out of heart attack every day. I mean, that's not really very impressive. Okay, but and most grants start, it's a waste of a sentence as far as I'm concerned. So if you tell me exactly where the state of the art, and then now you're making this, it could be a very small, there's nothing to be insecure about. It is supposed to be small but important. So as long as you clearly and sharply define it, I think you impress people and you score points.
I noticed a lot of people will start off and they get confused with what's significance in the introduction. An introduction kind of goes over what the state of the research is, but the question that you need to answer with that is, if my research is correct, or if it works in the way that I expect it to work, if my hypothesis is correct, will this have a significant impact on this field? That's what the question is. It's not, is the field significant? It's like, I'm studying, some childhood disease that, is horrible. We know that's significant, but how will your research have a significant impact on that field is the question they're asking. And innovation, remember, innovation is how you interpret it. In order to address that, I would start with looking it up on Google and seeing what innovation really means, and then trying to figure out how that applies to your research. So it can be technical, but technology doesn't answer questions. It allows you to answer questions, and you shouldn't only ask questions because you have the technology. You should develop the technology to answer your question. And I know those things sound subtle, but that's exactly what you need to really think clearly about as you write that down.
And anybody interested in DoD grants, I would advise you to be very familiar with TRLs, technology readiness levels. You have from 1 to 9, and there are certain grant mechanisms that will only accept you if you have crossed 5 or if you're below 5. So just look up TRL and then get some help to assess what TRL your science is now in. Typically it's developmental, and so if it is up to 4, 5 is a borderline case. With 6, you have really crossed over and you're now heading the market. So you really need to know. So the TRL 1 to 9 must be thoroughly discussed in a team setting to see what people think. And based on that, not only do you declare your TRL in the proposal, but also you justify why you feel that is the case.
In innovation, what I remember our SRO was trying to get us to spread scores and was saying, basically, if you're giving it a 1, how does this really change the field? How does this move the field forward? Everything doesn't have to change the field, but clearly how would this move the field forward is one of the key questions for innovation. Why is this new and different, and why should we care about it?
Just one little thing that I sorry, I had that set so I know when I was talking too long. One thing that's sort of come up with a few grant reviews of some of our young faculty members is where they take work that they've done before and then try to fill in gaps. They've published things, but maybe there's some aspect of it that wasn't really done that well, or maybe wasn't done at all. They don't really like you to fill in gaps. They like you to move things forward. So innovation really is to take what you have and go forward in a direction that science has taken you, not just finally complete what you've done before. If that makes sense to people, you all know your own projects, so don't be incremental and don't fill in gaps would be my advice.
Maybe a final comment: if you are an ESI and going to NIH, you should consider not to do multi-PI. If you did a multi-PI with another non-ESI, then your ESI status goes away. If you do a multi-PI with an ESI, then both ESI become non-ESI after that one grant is funded. So it is a deliberate decision. I'm not saying don't do it or do it, but there is something that you have to think about. If you're a non-ESI, I would strongly encourage have the multi-PIs you need to get the work done. Those days are going away where the Smith lab or this lab or that lab has to do it. I mean, it's about the problem. You have to solve the problem. And whatever talent sets are needed to solve the problem, you have to bring them to the table. And people do know that when somebody accepts to be a PI with you, they're stepping up the game and they're making significant room for this particular project in their sort of direct line of work. So multi-PI is encouraged wherever they're needed. For ESI, those considerations I said should be observed.
Well, thank you very much for all of your advice, and thank you all for attending.
Thank you.